color changes, bubbling reactions. Bacteria revealed. Welcome to Biochemical Tests.
Looking through a microscope will only get you so far. You can observe the shape and the stain of a bacterial sample, but when you are staring at an ambiguous colony like this, visual observation hits a wall. To figure out exactly what organism is growing on the dish, we have to map its internal chemistry. We do that by tracking its metabolic pathways, feeding it specific sugars and substrates, and observing exactly what it consumes and what byproducts it leaves behind. For that, we use a specific series of biochemical tests. We start with the gold standard rapid screens, then move into the 4-part IMVIC panel. Before you begin, you need a strict setup. You must start with a pure bacterial culture. You also need a specific set of test media broths and the exact chemical reagents required to trigger the final color reactions. If you follow this workflow precisely, you can turn a questionable swab from a patient into a definitive, actionable diagnosis. Step 1 is the rapid screens. We begin with the Catalase Test. Take a glass slide, smear a sample of your bacterial colony onto it, and add a few drops of hydrogen peroxide. If the organism produces the catalase enzyme, you'll see an immediate reaction. The enzyme tears the hydrogen peroxide apart, causing a sudden, violent effervescence. If you apply the drops and the liquid sits flat with a complete lack of bubbles, the enzyme is entirely absent, yielding a negative result. Next is the oxibacterial colony directly onto a specialized oxidase reagent disc, which contains a specific electron donor compound. If the bacteria produces the oxidase enzyme, it will oxidize the reagent. You will see a rapid, unmistakable color shift, turning the sample a deep purple within a matter of seconds. These instant screen reactions act as a crucial first filter. They immediately split your bacterial candidates into broad categories, narrowing the field before we start the longer incubation tests. Now we move into a deeper metabolic investigation, the IMVC sequence. This requires precise aseptic inoculation of 3 specific types of test media. Using a sterilized loop, you will transfer your pure bacteria into a tryptophan broth, then into a shared MRVP broth, and finally use a gentle zigzag motion to streak the surface of a Simmons citrate agar slant. Stop right here. Once the tubes are inoculated, you must not add any of your chemical color reagents yet. This timeline visualizes the required metabolic weight. The bacteria need a strict 24 to 48 hours in an incubator at 37 degrees Celsius. They need this time to actively consume the sugars in the media and alter the internal chemistry of the broths. Patience is non-negotiable. Rushing to add the indicator reagents before the bacteria fully metabolize the media guarantees false negatives, ruining your diagnostic profile. Once the incubation period is complete, take your tubes out of the incubator. We will read the tryptophan broth first for the Indole test. You are testing to see if the bacteria produced the enzyme tryptophanase to break down the amino acid. Carefully add a few drops of Kovacs reagent so they run down the inside wall of the tube. If the bacteria metabolize the tryptophan, it yields indole. The reagent reacts with that indole to form a distinct floating red ring at the surface. If your bacteria cannot metabolize tryptophan, the original yellow broth color remains completely unchanged, with no ring formation at all. Next, shift your focus to the Simmons citrate agar slant. This tests whether the bacteria can survive using citrate as its sole carbon source. The agar starts off a deep forest green, representing a negative result. If the bacteria utilizes the citrate, it yields alkaline byproducts. This raises the pH, triggering the bromothiamyl blue indicator and turning the agar a striking Prussian blue. These color shifts are direct biological proof. They allow you to visualize the presence of invisible enzymes and confirm an organism's ability to survive in a restrictive environment. Finally, we navigate the shared MRVP broth. Your first physical step is to divide that single mixture evenly into 2 separate test tubes. This shared broth tests for 2 different glucose fermentation pathways. The first tube is for the methyl red, or MR test. Simply add a few drops of the methyl red indicator directly to the broth. If the bacteria ferments glucose through the mixed acid pathway, it creates a highly acidic environment. The indicator detects this low pH and immediately shifts the broth to a vibrant red. The second tube is for the Voges Proscour, or VP test. Here, we are looking for a neutral byproduct called acetone. Add 5% alpha-naphthol, followed by 40% potassium hydroxide. You must wait up to 15 minutes before reading this tube. The chemical reaction to detect acetone takes time to develop, and reading it too early will result in a false negative. A VP-positive result reveals itself as the delayed development of a red or pink color, confirming the organism uses the butylene glycol pathway. This diagram illustrates the seesaw rule of the MRVP tests. Because these are competing metabolic pathways, organisms are almost always positive for one and negative for the other. They are mutually exclusive. Decoding the MRVP tests confirms the exact biological strategy the bacteria uses to process glucose. With that, your raw data gathering phase is complete. Now it is time to aggregate your individual test observations into a single 4-part IMVIC profile sequence. Let's look at a classic example on this scorecard. Say your results are indole positive, MR positive, VP negative, and citrate negative. This exact string of metabolic data points, plus, plus, minus, minus, perfectly matches the biological signature of E. coli. Take your unique scorecard and consult a standard diagnostic index, like Burgie's manual. Track your specific sequence of pluses and minuses until you find the corresponding pathogen. By reading these metabolic pathways, you have translated an ambiguous biological sample into definitive data. You now have the exact identity of the infectious agent.